Thank you. And that's it. So my first time. Thank you for the invitation. It's great to be here. It's my first time in Korea. Just all the way to KAIST. So I've heard a lot of good story about KAIST. So I know a few other faculties in KAIST, but finally I know somebody in the math department of KAIST. I know several people in the bioengineering here at KAIST. I'm a mathematician. I trained as a mathematician. I used to working on PDES, fluid mechanics. For the last about 12, 15 years, I started working on biology. So this is, I don't know, many of you has read this book. So it's by the National Academy of the United States and talking about the mathematical science in 2025. So they emphasize the importance of everyday life has used mathematical sciences, right? And they also emphasize the discipline of math as an overall scientific discipline is excellent, can integrate with many areas of science and engineering, and they also emphasize the training of mathematics. So I want to emphasize here is all parts of mathematical sciences. I think in this department there's a pure math, a lot of applied math, even within applied math, there are many areas. And they emphasize connect to a full spectrum of mathematical sciences. So, so you can see this is quite different from such a book before this one. This really emphasizes the applications of mathematics. So on the other side, in 2009, there's a book called The New Biology. This is also produced by National Research Council and the National Academies of the United States. There the emphasis is the new biology is integration or reintegration of many disciplines, including physicists, chemists, engineer, and mathematician. So in this country, in this university, I already noticed there's an institute called the Convergent Institute, right? Yeah. So in many ways, convergence means no longer divided math and science into many sub-disciplines. It's the integration of them to do the best possible research. Okay, so then I will show you a mathematician point of view of life, right? Of biology, life, right? So to me, the biology is a multi-scale system. It's really complicated multi-scale system. There's many, many, many elements, right? So in spatial temporal scales, okay? So that's one. Another unique feature of biology is self-regeneration, okay? So we have strong capability as a human, or a lot of animals can self-repair ourselves, right? And then we have this point of evolutions. So it's changing, okay? So our neck will get stronger because we use a lot of phone and use a lot of computers, right? And we're getting bigger. For example, recently there's a discovery. The stress on the parents can potentially pass. The stress can be passed to next generation. That stress can get the kids bigger and fatter. Okay? So I actually study this nowadays. It's called an epigenetic inheritance. Right? So the mechanism is different from a physical system. So most of us who working on physical systems, like mechanical systems, we are used to physical laws, right? So we're used to principles. So it means most of the time, the challenge is we know the principle. We use the principle to build a machine. The challenge is to build that machine, okay? But the principle is there. So so basically, I call this the white box approach. In biology, it's opposite. Most of the time, we know its function. We don't know how to build. Okay? So, for example, our brain works so well. And we know our brain can do a lot. But let me tell the truth, we know little, very little about how our brain functions. We know there's neurons, how they interact, how they function so well, okay? To memorize, okay? To understand it. It's still don't know, right? So then the system is complicated, really, really complicated. And how we study? Because it's a black box. So in engineer, we know we build things with piece by piece. Here, we're already here. So how are we going to study it? A typical way in biology is to take away things. For example, they knock out a gene. No, not a human. They knock out a gene in the mouse. Oh, how they behave. Or if they knock out huge change, that gene means must be important. If you look at it, this is sounds great. It potentially to be very, very misleading. So let me give you an example. You're driving a car. Okay? If you cut the wire between your key to the engine, you cannot even move. Now, if you're using the biology thinking, the most important thing, because the car originally can move, you cut the wire, you cannot even move. So that's why must be most important thing of the car. We all know that that's not the most important thing, right? The most important thing is the engine. So it means it's very hard to understand this thing. It's a black box, okay? So I'm joking, if in a math department or in a department, how do you figure out this one particular faculty is the most important faculty? So one way to figure, you delete them, means he will not come to the department for half a year. You see any changes in the department. If the change is dramatic, that's very important faculties. Otherwise, okay, it's just one of other faculties, right? So, all right. So biological system is very complex. Look at, this is a metabolic pathway. Means each dot is really already measured. Already people studied protein or transcription factor, some in the cell. Okay? So it's really complicated. So this is already complicated. Each of this is an element, they link with each other. Another unique feature is the variability. When people all look like different, there's no two identical human being, right? There's no two identical cells. So this is an example. No two identical cells. So let's come back to other side, is how to study it. Now, because the system is multi-scale, then it's starting from, this is structure of protein. This is the sequencing, genomics. People now, they know, this is sequencing. This is we have about 20,000 genes. We're sequencing it, right? And then this is a look at the cells, individual cells. Then this is a look at the fish, means look at the animal, look at the whole animals. So there's many, many data. However, you never be able to, at least current technology, say, at this moment, I'm going to look at the protein. No, you have to kill the fish, get the cell out to look at the proteins. That means many of the data, even they collected, they're disjointed. Okay? That means we have to connect. For example, one of the systems I'm looking at is looking at stem cells. You look at stem cells, so here's the cells. The cell talk to each other. The cell put in a tissue. The cell communicates. There's networks within the cell. At the gene level, there's genes, there's epigenetic information, state. They all talk with each other. How you can look at it? So the challenge is to how to link those data. Okay, so that's one of the goal of the mathematician can do. And also this is so complicated, spatial temporal dynamics. When things get complicated, so what do? Your brain just cannot wrap it. Then you write the equations. So before you write the equations, you have to write the right equations. Then you build a model, right? So when we, for example, from a human point of view, the engineering object, pretty much a passenger airplane is almost the most complicated thing. Okay? Usually an airplane takes about 20 years to build. From design, from everything. Now, then you say, okay, In order to understand that, one way to accelerate is build a model. This is a computer simulation model. Okay? So, you should build a model. Then what is a model? So, for many of us, this is a model, right? So, model is a way to show you the importance, how this, so this is the model for the clothes, for beautiful clothes, right? Now, if you really look at a model, so model, people has a perception about a model. So model basically, people tend to think a model is a description of the original object. That's not true. Model is abstraction of the original object. That's the whole point of having a model. If the model is already as the same complexity as the original one, then you don't need a model, right? So then the model are incomplete representation of the real world. For example, this is in the Museum of Science at Boston. There's a model of this, okay? And then you build this, you can understand how it jumps. Now, when you build that, if what you want to know is how this animal jumps, you really don't care that its brain, right? You just build the mechanics, how that works. So then the question is how this jumps when we build a model. So the models, there's many sorts of models. Okay? That's where the mathematics come from. So, for example, this is you can look at this coordinate system. This side is most complicated. Means you just put as many terms as you want, and as many terms as it needs, as many terms close to the reality. So, for example, this is the interaction of many genes. Well, but I don't know, there's 40, 60 genes here. And also the y-axis, this is magnetic, means I really want to understand the physical structure of the protein. And then this is the function, conceptually, which means I only care what the protein is doing. So in this symbol, the protein is degradation. The protein eats something. It's a classic notation. Our mathematician tends to think about it in a very abstract way, simple way. We tend to be here, right? So we tend to think about, it's 2 components, 3 components. We really want to understand it. We really understand it in great details. So all sorts of models are important, are useful. It's all about what's your questions. Okay? So reality is never here. Reality is over there. Right? It's really, really complicated, but you want to simplify it to understand. Okay? So today I'm going to use examples based on my own research to illustrate the point how to use a mathematical model and to understand biology. Okay? One question I'm going to address is positive role of noise. Means noise, that's your good, the good things. Another thing today, this morning I gave a class lecture in the dynamic system, nonlinear dynamics class. I emphasize how to get rid of noise effect. Now I'm talking about how to use noise, because you cannot get rid of noise all the way. Okay? When you cannot get rid of it, life is very smart. They found a way to use it. Another way is I'm going to talk about the stem cells. Okay, so let's get a simple model. Think about a model, think about questions. This is a fish hindbrain. Okay, zebrafish. Hindbrain means basically the back of our brain here. So we have some segments here. So in early embryo, this embryo that eventually develops segments like this. This color means gene expression. Means that a gene going to turn on. So the name is not important. Okay, the color represents that genes. All right. How do you pattern? This is the classic developmental biology question. So people say, this pattern is just like that pattern. So that pattern, if you realize this is a French flag, right? So we say, okay, how to pattern French flag? This is easy. Then all you need is put a molecules outside of the domain to give a diffusion gradient. Okay? If this gradient is very high, then they turn on this gene called the blue gene. Okay? Then it's very low because it's inhibition. Then too low, they cannot inhibit, then it turns on red gene. In between, doing nothing. Then you have 3 genes. then you have 3 different colors, then you patent. So this is a beautiful theory, okay? However, until about 15 years ago, this is a theory started in 60s, late 60s. You can imagine who will use the French flag as example to talk about the biology. Must be British, okay? French will not use the French flag to talk about, okay? So this is a British developmental biologist, right? So then the huge debating, where's this gradient? People believe there's a gradient. When they believe, they will not be able to see it. Only in now it's more like 20 years ago, late 90s, people started to see the gradient. Okay. At that time, I'm still working on mechanics, turbulence. Right? So then after people see it, the next question is, what caused the gradient? Huh? So we read the paper in 2002. It's all about the model. We explain the data. Basically, there's a bunch of data. There's a huge debate and people say it's impossible for certain mechanism to happen to get that gradient like that. This kind of beautiful gradient. This is space, right? Different curve means different time. This is simulations, okay? Simple PDEs, one-dimensional PDE, very easy to solve, at different time. Then you gradually reach a state. People say, it's impossible for diffusion to. Then we wrote this paper say, if you count right elements, you pick up the right size of parameters, you can use diffusion. Even our title says, do morphogen gradient arise by diffusion? Our answer is yes. Okay, this is our title of the paper. Okay, so this is my first biology paper ever. We're very lucky. There's no experiments, and it's a very good journal. And I said, okay. Then probably people still has many people say, if you believe it's diffusion, can you measure? So people here, most people might heard of diffusion, right? Just go measure diffusion. And we say, okay, we're going to measure it. So we worked very hard, but apparently it's very difficult to measure it. So we spent the next how many years? This is published in 2012. So 10 years, okay? This is the whole PhD thesis for one student. The 6th year is measure this diffusion. Okay, so this is the person. Right, it's the poor students, yeah. But he did a great job, and we measured it. So we have a model, we fit the data, we understand the data, we look at, okay. So here I'm telling you is, in both work, we use a lot of simple PDE continuum reaction diffusion equations. So I don't want to write the equations, okay. So it's very, once I write the equations, it's simple. Now, the key is how to write the equations. Then the story is finished, right? No, now it's complicated. Let me make it more complicated. So when we talk about French flag theory, that's really a beautiful theoretical idea. The gradient is beautifully like this, right? Then you turn on, then you have a border. Then this part of the cell is red, and that side part of the cell is blue. Boom, you're done. It's impossible. We know in real life, the gradient is more like this. Then you're going to have something like this. Means there's a region. It can be high or low. So basically, the color can change. Oops. That means something must be there you have to do. Then your next question will be, this is a beautiful theory too, but it's a theory, right? Did you actually see there's a rough boundary? Then we go measure it, right? You measure it, you see, look at each dot, it is a hindbrain fish. It's a fish, hindbrain. This is 11 hour after fertilization, after the life of that fish. 12 hour after the life. There's a 3-time image. Okay? If you look at it, the two colors means two different gene expressions. You can see this boundary is very rough. Both genes are expressed at certain domains. But later get perfectly shaped. So basically says, you have this domain, but this domain somehow fixed later. Now this is the observation, means you experiment, you can quantify. You can even quantify using image analysis, you just cut a line, you cut many lines, you see many curves. For perfectly, should be a step function, right? It's not. It's not gradually. Okay. Then we started this. Then I'm going to show a little bit of equations. So how can I start it? So I'm zooming in, okay? This circle is a cell. In the cell, inside the cell, there's a few elements. There's a gene here. They talk with each other, and there's a noise here. There's an external gradient. Of course, there's a noise here. There's noise here. No noise, of course, everywhere. Right? So you write the equation. So this part of the equation is ODE, gene 1, gene 2. And then this part of gradient is outside of the cell, is space. They're going to diffuse. They're going to take it in and out. So that's the equation. And of course, there's some stochastic terms there. Okay? Yeah, you can make it a color noise. And then you can analyze this. You can analyze this. Once analyze it means mathematical. And well, it's not that hard. It's not that easy if you have PDEs. But if it's ODE, if I say no space, then you can do something. You can use the Windsor-Friedin theory. You can use minimum action path to estimate the transition from state to state. So you can do, this is really a dynamic system with 4 or 5 stable states, 6, 7 unstable states. You can analyze it, okay? Then you can estimate switching probability from one state to the other state, use large deviation theorem. So you can do a lot of this, but this beautiful theory, it only works under condition there's no space there. Okay, once with space, yeah, you still can give you some hints, then we just simulate. The stochastic PDE, we just compute. Okay, so let's do the computation. Let's do a computation now. So the nice thing about modeling here is we can control noise. We can turn it off, we can add it, we can make it bigger, we can make it smaller. In reality, you cannot do it. So here what we do is we do simulations. Let's just look at these pictures. Each square is a cell. So we put the noise where? The noise is in the morphogen. The noise basically is here. We put it in. We got this very rough boundary. And that's not surprised because you have noise, the boundary must be rough. Then the question becomes how they get it sharp, right? But another noise I haven't added in is this noise, the gene expression noise. The next thing we add is basically the noise here, the dynamic system noise. I haven't added it yet because I set this equal to 0. So here's the amazing thing. We add that noise, then the boundary gets roughed, then it gets sharped. So I get this result when Lei Zhang get this result. So I said, you're wrong. You must be wrong. How could you have more noise than it gets sharped? She says, yes, I'm wrong. He go check. I still get this. And then we spend about 3 or 4 months. We say, well, there might be the reason it's not wrong. Then we look at the more. We did a lot of analysis. ODE analysis means stochastic dynamic system without space. So here's the explanation. Instead of giving you all the theorems, okay? So here's the explanation. So most of you, I don't know, some of you at least knows this bistable behavior, right? It's a bistable. So the noise outside is the input. You think about the signal. And to get the downstream gene expression, they got 2 colors. The color is responsible by the downstream response, which is bistable. So in this middle range, there's some cell which is blue and red. Blue and red. Okay. If you use the energy landscape theory to think, it's in this region, there's 2 wheels. One is blue, one is red. So what noise is doing? Noise, you think about what the noise is doing. Noise basically is just shake this. Or shake this. If you shake this, of course, what do you mean by shake it? Well, it's a fluctuations. So once in a while, if you're big enough, boom, they make this jump. Gradually, you lose most of the blue cell in the shallow well, went into the deep well. You can analyze this mathematically. So that's the reason. Because the noise-induced switching gets you from one state to another state. For example, if I'm standing here, if you keep pushing me once in a while, I'm going to jump to other states. Okay? All right. Now, but the key is, so this way has to be, one way has to be shallow, one way has to be steep. If it is two ways as equally steep, you're in trouble. Because the equal cell come to here and go into there. Then you just change colors. It still doesn't work. Now, a lot of simulation here, and then we worked hard to develop numerical method, right? So, if some of you come to my talk tomorrow, I will talk about more of this. The idea is, in biology, all the chemical reactions, the biochemical reactions in this nonlinear term F is very stiff. When you're very stiff, then you need very, very little time step delta t to capture. Why? It's like this, right? It's like if our hands move very slowly, that's one time scale. What kind of delta t you need to capture my time scale is basically that time scale per second. Right? Now, even my finger, watch my finger. In the meantime, my finger goes very fast, suppose. You write this whole thing as one dynamic system. Then that time scale is going to be the dominant one for your delta t. That's terrible. In this case, chemical reactions, the fold of difference can be 10 to the 6. Sometimes it's 10 to the 6, faster or slower. On the other hand, suppose you really don't care about this time scale. Means you really don't care how my finger is shaking. You all care my arm is moving. Then you wasted it, okay? So that's what we're talking about here, okay? Now, of course, we add stochastic terms, blah, blah, blah. That's one. Another thing, I know you guys here has a lot of effort working on stochastic dynamics, right? I show this probability density function. If you write a kind of probability function, the most challenging one in terms of computing it, even analytically, is when this x becomes more than 2. Now, if this x is 2 or 3, you can almost link to the physical system, the space, x, y, z. Now, in biology, this x is the genes. Is this gene, A gene, B gene, C gene, D, F, E, we have 20-some thousand genes. If you only take 10, oops, this becomes a dimensional 10 problems. How to compute it? Well, in the space, we use something called sparse grid. Use time, we use something similar to the previous papers. So we compute all the way to dimension 6. Okay? It's already getting very slow. All right, so I detoured to the biology, this is all computational method. For students who are really interested in computation method, you can come to my talk tomorrow. So I'm going to talk about more of this. Now, on the biology side, if you're a biologist, then you care is, you have a beautiful theory, using noise. Tell me, where's the noise? Can you measure noise? Right, then, after getting that, we literally true. After we published that paper in 2012 with Lei Zhang, then we write a grant to measure the noise. Okay, so it takes us about 2 years to get the grant. So 2014, we got the grant, we measure, and then we model. So each dot is a cell. Then there's many animals. Means we killed a few fishes, right? And then measure the fish, the few cells in space. This is the micron, right? So we found, amazingly, you can control the level of noise, means the fluctuation level, by control some of the genes. Okay? So we said, wow. Then we still don't know why you control some of the genes. You look at this. From here to here, this is the normal case. From here to here is the mean stay about the same. The fluctuation gets bigger. From here to here is the mean get changed, but the fluctuation get the difference. So that means in this system, almost as an independent control, means independent regulation, can go through these two genes to control mean and variance. Okay? So noise is good, right? Have you believed the noise is good? Means I can use one noise to battle the other noise. Means this noise can induce switching to correct the mistakes caused by the other noise. Right? All right. So you ask yourself, is this a general principle? So let's study another problem. Okay? So this is a little bit even more easier to understand. This is the earliest part of the life. Okay? Mammalian means for our human, it's just like this. We're starting from 1 cell, 2 cells, 4 cells. Gradually, we're getting here. E45 days means E4.5 means after fertilized, 4.5 days. You're getting this. Of course, this is mouse, okay? It's much, well, I have pictures for humans, but it's not good to talk about human. But this process is very similar. People are studying this. It's one of the hardest topics. Right? Now, for example, we even proposed how to control this to do test tube fertilization. It's already done, right? But the test tube fertilization has a very low successful rate. So how do you improve the rate? Okay, so one major question in this problem is, goes from the here, you say 1 cell, 2 cell. They must be the same, right? 2 cell must be the parent cell, and the 4 cell, okay. But people knows up to this stage, have shape like this. This type of cell and the inside of a cell will be totally different. So this type of cell, these cells will eventually become your guts, your body, or your stem cell. This is called ICM. This is become the embryo. The outside of embryo eventually will be disappeared. And then this part later will also divide it into 2 types. Some eventually become your blood, sometimes becomes your other part of organs, like skins, which are fundamentally different. Blood cells, skins are fundamentally different. So our human have about 200 some types of cells, okay? So the question here is, how did you get the inside outside? We need to be in the right location and the right fate of the cell. Means the color has to be right, the gene has to be right, okay? So my usual question is, how did you guys know if I'm sitting here? Cells don't have an eye, right? They can only feel. How do you know you are sitting inside or outside? Right? Some of the students sitting inside, some students sitting outside. How did you know? You have no eyes. Suppose you close your eye, you cannot talk. How did you know? You can feel. This is how you feel, right? You touch around. If all touch around have a lot of chairs, I must be inside. If I touch around a lot of empty space, I must be outside. Most likely to be outside. So there's a theory called this, called inside-outside theory. So we come up with, but there's already some fundamental elements there. We just summarize it, okay, called inside. So here's the way. Happens if you say, this is great. Did you know the cell actually has a way to feel it? Yes, they have a gene called TET4. It's a gene, when you squeeze it, when you contact, this gene level goes down. This gene level goes down, and then this lets down, and then this up. This up means, the inside cell will contact more, then the color becomes stronger. Then the outside cell will not. Is that beautiful? Are you convinced? Now, okay. So if you're convinced, then it's okay. This is typically it's a short form model. It's a model in your head. When you think about it, it all makes sense, right? Then with your simulation, it makes sense. It makes sense. However, you have some mistakes here. Means some of the inside cell still have the expression level or gene expression of outside. If you think about it, it makes sense though, because the cell has to divide. Divide is a very random process. And then that means in this whole process, some other randomness will give you some errors. That's not surprised. Okay, so we play the same game. We go looking at the gene expressions in the cell. That's the experiments, okay? Each dot is a dot. It's really a cell from different animals, from many, many embryos. This, for this case, is mouse. It's really expensive. You kill one mouse, maybe $500 or couple of $100. You kill another one, you kill another one. Okay, you kill many of this because you have to make this mouse in a special way. Then you found there's a fluctuation level for ACT, okay, in this way. And CDX, different levels in this way. So there's fluctuations. And then we add this noise in the system. So it's 1 cell, 2 cell, 3 cell, 4 cell, the colors. Then you're looking at it. So, I shouldn't push this. Once I push this, they stop. You can see, right? Some of the blue cell change the color. Because of what? The only difference between this and that is it has noise in the genes. If you, yes, you can look at it. So we can add the noise. You can measure the noise. The noise is for the blue gene. This is real. This is not simulation. This is real data. The blue gene about 30% coefficient of variation. The red gene is about 15%. We actually add that number in the system. We see this beautiful. And then you see more noise. This is corresponding to different noise level. More noise. This is corrections. Means the fraction of the correctness. 1 is 100%. You can see noise bigger is improved. But I cannot get it too bigger. If it's too big of noise, it comes down. 30-40% give us the beautiful. You can do this even more for 3D. Do you know why? 3D, 2D, there's a fundamental difference. If you are a geometry, you say, for 2D, your neighbor means what? It's a plane. This is my neighbor. For 3D, neighbors everywhere, right? So you have more neighbors, actually, roughly speaking, relative to your surface area or your arc length. So we do the 3D. The 3D, actually, the correction is even better. Means the noise level. How to do 3D? Then we can even do more later. Now this work only up to this stage. Now we're expanding the genes. We're going to up to here. So we have to then get complicated. This is basically the computational domain. So basically very complicated mechanics and biochemical reaction. So this is the model we have molecular as well as mechanical interactions. So I hope then this mechanism is the same thing. The contact introduces a loss level due to division. The gene expression makes the switching to correct the error. Same as before. The before is the errors introduced by morphogen gradient. The gene expression correct the error. We have even more example like this. Okay, so now let's switch the gear. Let's do something. Let's look at something different. Stem cells. Okay, so the first part is a story about the positive role of noise. The next is I'm looking at stem cells. How much time I have? 20 minutes? Okay. So, stem cell. So what is stem cell? Stem cell is very useful in many parts of your body. People tend to think about stem cell is only for development. No. The skin, our skin is regenerate about 7 to 10 days. Maybe it's 7 to 14 days. It's a wide range, okay. This is the crypt, the colon crypt. The regeneration about 7 days. It means 7 days, some of those cells will disappear. Of course, in a random way, but average. Those are stem cells. They're going to generate this blue cell. The blue cell is going to generate that cell. Then the constant changing. If you think about this, wow, in your whole life, some of this cell will stay there forever or reproduce themselves. This process will keep it. So you think about it. This is a magic. Did you have any other mechanical sense you can reproduce the self in that way with a perfect, right? Okay. So, what is stem cell? The stem cell has two properties, self-renew and potency. Okay? So self-renew is they produce themselves. Potency this means that they can produce other type of cells. So how to think about this mathematically? Right? So we developed a model. I use the word universal, means I really don't care. This is this stem cell, skin stem cell, colon stem cell, or blood. They all have these structures. Is this stem cell going to produce themselves and produce other cell? The other cell, some intermediate cell, eventually become a permanent cell. Okay? How do you write the equations? It's not hard. You just say, okay, there's a certain rate, you divide it. There's a probability to become this type and that type, all back. 2 cells, okay? You're going to lose 1. And then this is the time constant related to cell cycle. This is the number of cells. We use concentration. And you can write equations. The reason we write this dependence because they have potential feedback. So all the probability of reproduce themselves has certain probabilities depends on the number of cells. That's it. Now you can analyze this. You can, you can analyze the number of state. You can do linear stability. The most important, you can do global stabilities. You can characterize. You can show. So that I will not get into it. So the interesting is the negative feedback has a way to accelerate the regeneration. Okay, that's what we found. Means you look at 2 curves, right? We're talking about. This is a very specific system related to olfactory and epithelium. Means your sensory system. You are here. You can sense, right? There's a neuron on top. You get a cold. You lose a lot of neuron. If you lose neuron, your system is very smart. They're going to regenerate. Usually, it takes like a couple of days to regenerate. When you have a bad cold, you cannot smell anything. So that means you lost a lot of neuron. So you want your regeneration is like that fast. You don't want your regeneration like this. But fortunately, for this type of feedback, you have this kind of regeneration. For this kind of feedback, you have that kind of regeneration. Because this is cell cycle, this is related to the probability. You can do a calculation. You can analyze even by linear stability. Okay? So, then you say, how did you get the feedback? It's also like a magic. There's feedback to that. Then what do you mean? Means I do, as a cell, I do something. I will tell Jay to do something. So what am I going to do? I cannot talk. So one way to do is I release some molecules. The molecule is going to go there, touch him, say, I have 10 molecules. And you say, don't do much. I have 5 molecules. You can do a lot. Okay? Now, you can use that molecule. We can use that we can test the molecules, right? And we also test many other type of feedback to looking at the accelerations. Okay? So, with this fundamental framework, we can study something which is more useful, the breast cancer. Okay, so this data motivated our study. So if you look at this is just a bunch of cells. It's not interesting. What is interesting is this number. So what do you see, the number? The number is increasing and decreasing. Increase, then decrease. That number is a percentage of the cell, which happens to be stem cell. Use this marker. So it's cancer stem cell. Basically, the cancer stem cell increased, then decreased, percentage-wise. So when you get this data, you're very confused. I'm not confused. The biologists are confused. They're confused. They say, how could stem cell increase? Because they know The stem cell will not die.
It should then keep increasing.
Then if not die, then it should keep increasing. Or maintain it. Then we say, okay, if decreasing, then means you must have negative feedback. Then we introduced a negative feedback. But we don't know where the part of negative feedback should go. We just put all of them. So then that's a nice thing about the model, right? You can explore it, you know. We will not kill any animals, you know. It doesn't cost that. It just costs the computer time to add another feedback. So we just add it, and then we can fit the data, right? And then only when you add all this stuff, then you can actually find that it works well. Means you can fit well. Now, we try to look into more. The next question is, okay, feedback. Which molecule does the feedback? The biology is always clear, you know, if you can argue there's feedback. I said there must be a feedback. The question is how the feedback works. Which molecule does the feedback? We're still working on it. Apparently it's not easy. I thought it's easy, but it's not easy. We've worked like 2-3 years now. We still haven't been identified the feedback molecules. But we can do something more useful to use this as a strategy to thinking about therapy. Okay? So, because you have cancer stem cells, you have other cells, when you're doing therapy, there's many strategies. One is, I'm going to kill, who cares about this? I just kill the cells. So that's the what? That's the chemotherapy. You just kill the tumor. You try to kill cell, health cell, non-health cell, stem cell, not stem. I'm just kill it. But I'm focused in that part of your body, just go kill. So that's the strategy number 2. Another strategy is you give a drug. That drug is only targeted on the cancer stem cell. It's only targeted at some of the pathway related to cancer stem cells. Okay, that's one. Then you can perform not on human. Okay, this is the human data. This is the drug, very expensive drug. Okay, they have 21 patients. The dots they have a mark on this. Okay, right. So we do the simulations. The simulations, if you look at this, is tumor size. For tumors, if you put a drug, the doctor gonna go to watch after a few days. Oh, the tumor shrinked. They're happy. The drug works, right? But here is you look at the 3 curves. Up to here, you are happy with which curve? The red curve. Change the most. Okay. The drug 2. The drug 2, drug 1. Drug 2 clear is better than drug 1. And then the drug 1 plus 2 is, from here is not clear, is better than the drug 2. Okay, so that's the conclusion. Drug 2 is better. After 400 days, you have to stop your drug sometimes. You stop your chemo, then you found that the drug 2 is terrible. Okay, so this is looking at the volume of the tumor. You can also, to looking at the percentage of stem cells. In reality, that's very hard, almost impossible at this moment for humans. But for computation, of course, I can. Then you can find, wow, your strategy of this better strategy of drug 2, from the very beginning, the cancer stem cells increasing. Even though the whole tumor shrinked, the inside of the cancer is increased. If you think about this, not surprised. Do you know why? If I remove a lot of this cell, the whole tumor is most occupied by this type of cell. If I remove this cell a lot, this feedback gets weaker. Then poops, this guy going to increase. This is increased, reproduce themselves. You have a lot of cancer stem cells. Then once it's exploded, then it become exploded. And then, this is you use chemotherapy, we can match the data. This is the other drug, we can match data. So, the idea is, it's even worse than not doing any treatment in our study. because you get rid of the inhibitor, because the other cell try to inhibit the cancer stem cells. So we also we actually can simulate the tumors in some spatial ways. If people are familiar with this moving boundary problems. So let me skip that. Yeah. So if you like this, this is my favorite topic, mathematical wise. Still one of my favorite is solving integral equations. This is my love. When I was a graduate student, that's what I do. I solve an integral equation. Okay. It's very complicated. So let's switch this to stem cell. So adult stem cell, in blood, there's many different cell type. One cell type is a blood stem cell. That is very important. Means ours, for example, if you donate your blood in certain portion, your blood will be regenerated quickly. Your health, you're fine. People can even do this. Because when you donate blood, you can only take out certain volume of your blood, not too much. But for example, if somebody needs a certain type of blood cell, I can do the following. I can get my blood out, I extract that cell type. That type I give to you. That blood come back to me. So I only extract that type of cells out. I donate that type of cell to you, a lot to you. You need it, but me, amazingly, the regeneration will happen. Somehow they're going to recover that percentage. If you think about it, this is amazing. How they going to blood achieve this? The same thing for the hair. We lose hair. I care about my hair. I'm losing my hair. So we lose the hair. Most of it is the stem cell. There's a stem cell here, few stem cells regenerate. Okay. How you think about this? So we think about this in a optimal control setup. Means we have objective. Means from one generation cell to other generation, my emphasis is looking at the cell the distribution of different type of cells remain the same. So it's a totally different question, right? It's not about the number anymore. It's the number and the distribution. Okay? So then we have something called X. X is genetic factors. So you can model this as optimal control. You maximize the performance. We go backwards. We say, suppose the goal of the whole system is at this cell generation, one cell, one cell cycle, the population distribution in certain molecules, it has this distribution. After this cell cycle, they want to maintain this distribution. So the question, if under perturbation, how did you maintain this? Then if you maximize it, that's my performance. Expectation of that has to be in that form. How did you get that distribution? So that's the question. Basically, what kind of regulation you need? Apparently, this is related to economics problems. So I don't know, some of you can read this. This is about how do you distribute your wealth. So here's a classic economic problem. I think it's particularly true for Asians. Do you know why? Is at a certain age, you have a certain amount of wealth in your pocket, right? Your money, your house, everything. Now you have kids. The question becomes, what's the percentage of your welfare should go to the kids? So that's the economic questions. To maximize the happiness of both you and your kids. Apparently, this is a not easy question because there's a lot of uncertainties here. For example, most of us will not think this way, but that might happen. You might have another kid. Even at this moment, you say, I'm not going to have a kid, but you never know. You might have another kid. Okay, you might have another wife or another husband. You never know. That's one. Your kids might become very rich. The uncertainty, if he's rich, he doesn't need your money becomes epsilon. To him, relatively speaking, right? So, and the other way around, your kids might be very poor. They really, really need your money. So all this factor can process in, and of course the number of kids. So this can put a famous, so this is two famous, really famous people. One is famous economist. Another one is ecologist. They basically use ecologist theory working on economy. So this guy is a Nobel laureate. So that's not a surprise. He has 3 or 4 students as a Nobel laureate. Okay? So what I learned is, so they work together on this. I said, okay, we want to use this theory. I work on stem cell. He didn't work on stem cell. He come to visit our university. He said, you want to work on stem cell? He said, I believe stem cell has something with doing with this. He gave us these papers. It's about that time. So we look at it, we started. So what we learned is something almost trivial and almost very against the intuition. What we found is you need feedbacks. Okay? You need feedback, and also you need heterogeneity. Means each cell should have the different lifespan. That's apoptosis. Each cell, if they all die in 5 days, that's bad, that's the worst. You should have a distribution in order to keep that distribution. That's one. Okay? So it means I want to keep this distribution, you'd better to have a way to the cell. With each cell here responds to the death differently. That's apoptosis. And also, you want the feedback depends on the proliferations. Means homogeneous feedback is the worst. Means your production of yourself also should depend on where you in that distribution. Okay? So it means uniform is the worst. Okay? So a heterogeneous proliferation is also important. Okay? In particular, if you lose something, you want to regenerate yourself. Say, we can do simulation, we lose something. Heterogeneous proliferation means the proliferation depends on the spatial local. No, it's not spatial, means your epigenetic state. That's the most effective. And if you, in evolution, if you have cancer, evolution means during a lifetime, some of the genes are going to mutate. Okay, the mutate, you still want your distribution to be consistent. Then you better to have the heterogeneous regulations, the epigenetic heterogeneous regulation. Otherwise, you cannot fix the mutations. So everything indicates the best way is diversity. That's why you think about more people think about the Earth. How to maintain the Earth in a healthy way? You should have you make sure every animal should be still there and have the correct and distributions. So I'm getting close to finish. Okay, so we publish the papers and people like it. People say, oh, this is actually related to astrophysics. I still don't understand why this is related to astrophysics, but it's okay. So the next thing we say, okay, is we look at the skins. Right, this is wonderful. And there's different part of skin, different tissues. This is the skin disease. It's related to these bumps. Okay, that's the cell lineages. Really complicated equations. Now you talk about equations. Really complicated equations. It's a moving boundary, and there's reaction. There's lineages, and a lot of equations, and a lot of equations. And we have to, because this is a moving boundary, you have to do some transformation. You have to use multi-grid to solve it accurately because you want to compute the surface tension, which is the curvature. Related to curvature, this accurately. Curvature depends on second derivative. And then you Laplace another second derivative. You eventually this information also go back to Laplace, and you need to be high order. Okay. And then another way is we're looking at individual cells. We track individual cells, and so let me skip off this. I think just too much. Oh, this I want to show. So let me tell you, I really care about the hair. We really work on the hair of the mouse, right? So let me finish up by saying hair is very important. We tend to think for human, who cares about hair? Hair just for appearance, right? If we don't have a hair, then what? Now, this is our recent funding. So this is particularly good to talk about this in Korea. Wound healing. We found, using mouse, not a human yet, okay? There's possible to have wound healing work like regeneration. If you think about what do you mean by wound healing like regeneration? Wound healing, when you hit a wound, you're going to have scar most of the time. Regeneration is naturally your body is doing the work. It means nothing special going to come up. It's just as normal as before. So we found, interestingly, not we found, some other people and us, we found is, if you cut two wounds, one big, one small, in mouse, did you guess which wound heal better? Small wound, big wound. So intuition will be small wound. What we found is big wound. The reason is that it's big wound will feel better. What do we find? When heal better means it's almost like a totally recover. It's because they have the hair first. If the small wound, the middle will not have the hair before the heal. For the big wound, what we find is to have a hair first, then it started to. Somehow, the wound healing is like energy regeneration, has something to do with the hair there. Okay? So that's what we're working on. We have some simulations. Let me see if I can. This is 3D simulations. Let me see if I can. You can think that that's the wound. The middle is the wound, okay? For one case, you can heal it. For another case, you cannot heal it. All right. So let me finish up all the way to the end. This is two. Oh, another thing we do is, I don't know how many people here working on optimization. We working on optimization. Use optimization to working not on imaging. We're working on optimization on construct gene networks. Okay, so let me finish by saying this. The first part of talk is on noise. Okay, so now hopefully by hearing my talk, when you think about noise, you think about the possibility noise can do good thing. And what we talk about is how to use noise to correct the error introduced by another type of noise. And by looking at this, the mathematics can potentially produce a lot of new principles for biology. And that's the biology leads the most to me. There's infinitely many systems in biological systems. What they really lack is a general principle. But I believe, unlike mechanics systems, you have 3 laws, a few equations, you're done. It's all about how to figure out the boundary condition, different complicated geometries. Biology might need 20 laws or 200 laws, I don't know. There are a lot of laws. Okay. Also, their laws is much softer than, because the variability. It's not like exactly this equation. No, the equation plus some fluctuations for sure. Right? So, and I talk about a little bit the modeling and the development. And so those are the people who did the work related to this particular talk. Some of my former postdoc students. And those are the people who I never do experiments. Okay, so they do experiments. I work with them. And thank you for your attention. But I really want to say, I believe, I heard this is my first time to Korea. And then I also heard a lot of exciting things, in particular, applied math in general, finally might have a chance to take off in Korea as there's more emphasis from government. From KAIST, I can hear a lot of good news about the math biology. So I put in these slides and say best wish for Jay and all the math biology in KAIST, and hopefully you have a tremendously fast start in your success. Okay? And thank you for your attention.